prominent biology classes. Today we are going to discuss the separation and isolation of DNA fragments. In previous video we studied about the DNA fragments, how do we get the DNA fragments, and in this video the separation and isolation. Separation of DNA fragments is done by gel electrophorosis technique and isolation by elution. In gel electrophoresis, there is the word gel, electro and phorosis. That means here the gel is used, and the gel that is commonly used is agarogel. Agarogel, it is a natural polymer extracted from seaweeds. Electro means the current is required here, and phorosis means the movement of DNA. So under the influence of electric current, we can see the movement of DNA in the gel, and this is the scooping off, extracting the DNA fragments from the gel. Let's see the apparatus in which the gel electrophoresis is done. It is a glass tray, buffer tray, in which is filled with agarogel. And in this agarogels, the holes are made. These holes are called as jewel. And the fragmented DNA molecules are poured here through this hole. And when the electric current is supplied to this tray has a cathode end and anode end. When electric current is supplied to this tray, the DNA fragments moves. The larger fragments, they remains near the well, and the smaller fragments, they can move far away from this well. Now let's see the steps that is taking place in this technique. This is the overview. Now come to the steps. Let's start with the first step that takes place in gel electrophoresis. When DNA fragments are poured into this tray and there is a current, the current is flowed through this tray, the DNA fragments are separated. As we all know that DNA molecules are negatively charged, so they move towards the anode end. Naturally they move towards the anode end. In first step, the DNA fragments are separated according to their size, and the sieving effect is given by this agarogel. So size wise the fragments are separated. Means the larger fragments are nearer to the well and the smaller fragments moving far away from the well due to the concentration gradients. So in this way the fragments are separated. Now these separated fragments are stained with the stain, that is the Ethidium bromide stain. Why the staining is needed? Because the agarogel is a transparent and DNA fragments are also transparent. So to locate the DNA fragments, this staining is required. So after staining with Ethidium bromide, it is exposed to UV radiations, and in UV radiations the DNA fragments, it appears as a orange bands. So by identifying our gene of interest, we just extract the DNA fragments from this agarogel by the process of elution. Elution is just scooping off the DNA fragments just like an ice cream. So this elution is done. So in this way we can separate the DNA fragments and we can just isolate the DNA fragments also. So after isolation we have the purified DNA fragments, and this purified DNA fragments are used for recombinant DNA technology, and by joining them with the vector with the help of DNA ligase enzyme. So in the next step we need the vector for the cloning process. So in next video we will study about the cloning vector. Thank you for watching.